So the HLA class 1 antigen presentation machinery, there are lots of proteins involved in this. So the first, like for example, if there is any accumulation of aberrant or dysregulated protein, they process to the immunoproteasome subunit. That's basically a proteolysis process where the large bulky proteins get cleaved in small peptides, and they further processed through the endopeptidases. So from a bulky protein, they become very 20 to 30 amino acid. And then they stay in a cytoplasm. And then to load onto the MSC molecules, they need a whole set of which is called as the peptide loading complex, which is composed of lots of sub proteins. So TAP1, TAP2, they are the two major proteins. Basically, they are transporter proteins that allows basically transfer of those peptides from cytosol to the immune, where basically the MSC molecule is situated. So that's why it's very important. Now you can understand like the whole proteins are getting degraded there, and they are sitting in a cytoplasm. But if they are not able to go and bind to the MSC molecules, there is no meaning of this whole process. And for that, they need a very fine molecules like the proteins. In that case, TAP1 and TAP2, they are basically transporters. So they are the initial checkers for these lyse proteins. And then they check these proteins, and then they allow the entry of these short peptides from cytosol to the endoplasmic reticulum. And once they are entered, then they're further processed by different peptidase and different chaperones. They shape, they trim it, they tune it, and then they further load it to the MSC molecule. And with that, they assemble with the beta-2 microglobulin. That's a very critical protein. So with the beta-2 microglobulin and MSC, with the bound peptide, they go to the Golgi, and from the Golgi, they go to the surface of the tumor cell. And now it's there and displaying. So now the CD8 T cell will not, when it will be there, they will recognize those tumor antigens, and then they will start doing their function. But the problem is, if the peptide complex with the MSC is strong enough, still CD8 T cells are not able to induce that prominent response. There must be some problem there, because once you have the tumor antigens on the tumor cell surface, but tumor cells are smart too, like they will try to evade that thing. So they will try to evade, doing something with the antigen presentation machinery. They will try to disturb the whole machinery, so there is no presentation of those antigens to the surface. And then if the CD8 T cells are sitting there, they will not recognize those antigens, and nothing gonna happen. So our project, like we are trying to, we are proposing that TAP2, the one of the transporter protein in the TAP family, the TAP2, like so for the clinical purpose, like what we did in the study, so we have around 1,100 non-small cell lung cancer patients, and in a different cohorts. And then we have around 200 immunotherapy, that's naive immunotherapy treated non-small cell lung cancer patients. And then we screen the expression of TAP1 and TAP2 protein in those cohorts. So what we found that there were like around so many cases, which was around 45% cases out of those 1,100 non-immunotherapy, there was a frequent downregulation of TAP2 protein, which was 45%. It's a lot compared to the 5% loss in expression of TAP1 protein. So you can see like they both are responsible for transporting those peptides from cytosol to ER, but major defect was found in the expression of TAP2 protein compared to the TAP1. So that led this foundation to study more in depth about the TAP2 protein, because they both are responsible for transporting those peptides. But why is this so selective with the TAP2, not with the TAP1? And let's say like example, like in immunotherapy cases, like when the clinicians, they screen the patients, and when they design the immunotherapy for a particular patient, so the basic thing, like, or maybe you can say the major determining factor in designing an immunotherapy research or the treatment is to see the PD-L1, PD-1 expression, because that is like patients with having the high PD-L1 expression, they're gonna respond more to the PD-1 therapy. If the patient's having a low PD-L1, there is no meaning to give the PD-1 immunotherapy. So that's the clinical significance. So now you assume like you screen the patient having high PD-L1, and you start doing the immunotherapy by giving the PD-1 blocker, which is basically blocking the checkpoints on the CD8 T cells. But still, if tumor antigen is not on a surface, so the whole thing is not going to work. because if you block the checkpoints on a CD8 T cell, but still CD8 T cells need to go and bind to the tumor cells where the tumor antigens are there. If you have defect in the antigen presentation, and if you have no strong antigenic signal on the tumor surface, the whole functional CD8 T cells not going to work there. So our whole approach is to discover a good biomarker together with the PD-L1 or PD-1. So we are proposing that based on our data and what we have seen, that measured with the cases, like around 45% have defective TAP2. So if we design something where we screen patients based on the PD-L1 as well as the TAP2 expression, then that will be a very useful approach to design an immunotherapy for those patients. And so that's the clinical importance of this project. And also, like, we did lots of in vitro study to validate what's the signaling, like what is triggering and what is so selective for the TAP2, not for the TAP1. So we designed lots of in vitro experiments here. So my project is combined with the basic research as well as the clinical aspect of it as well. And the next part of this study is to screen some compounds, like the compounds which has the therapeutic importance. So now we find the problem. So the next part is to find a solution. Like, you just say, like, this is problems. Now the next thing is like what we can do now, like in clinics, like how you gonna improve the TAP2 expression. So the other part in this project is we are trying to screen some therapeutically relevant compounds. So we're gonna use those compounds in our lab scale first. So we have lots of model system, like we have the tissue culture system in our lab. We are doing organoid system. We have in vitro cell culture facility. So we are in collaboration with the Yale Center for Molecular Discovery. We are trying to screen some compound which are able to recapitulate or to enhance the expression of TAP2 in lung cancer patients, in lung cancer samples, and then we will, in the future, like, we can start trials with those compounds.